Okay, now we need to move a little bit more towards an organismal level. And what we want to consider now to ultimately understand the foundation for genetics is we need to understand what's called the gametic life cycle. So, like I said, as we move into genetics, we want to define some terms. So, we know that living organisms need to reproduce themselves. We know that if I'm a single-celled organism, cell division is itself the mechanism for reproduction. If I'm a multicellular organism, though, it can be a little bit more complicated. And what we're going to see is that genetics is the scientific study of heredity and variation. So what do you pass on to the next generation? Heredity is the transmission of traits from one generation to the next. And variation, then, is so important to Darwin's observations that we see that the next generation is different oftentimes than the parents. Differences in appearance that offspring show from parents and siblings, especially your siblings. So the fact that you have the same two parents with the same genetic information, yet you and your siblings aren't identical, says that there's something going on. And that something is sexual reproduction. So sexual reproduction is really, literally about all mixing up the genes. Mixing up the genes that you have to pass on to the next generation. So we're going to have an exchange of genetic material from two individuals for reproduction. And that individuals is in quotes because I'm going to use that loosely. As you study more biology, you'll see that that's a broad definition. Half of the genome of the progeny must come from each parent. All right. So this is going to beg the question then, going back to what is the structure of your genome. And ultimately we're going to see that you have two copies of every gene and that you are a diploid organism. And that you hand off one copy of every gene to your offspring. That's kind of the key to it. And then when each parent hands off one copy, you are returned to your status of having two copies of every gene. And that is called being diploid. So meiosis is going to be the process by which I take half of my genome, the one copy of every gene, and make a gamete from that. So that's going to be sperm or egg. The chromosome number is going to be halved. So humans have 46 chromosomes. So when we reproduce through the process of meiosis, we make sperm and egg, and those each have 23 chromosomes. Now you can see how this is essential, because were this not to be the case, then the chromosome number of your progeny, your offspring, will continue to double, and that will be unsustainable. All right. Now this isn't the only way to do it. Sexual reproduction in the gametic life cycle isn't the only way to do it. There are other sexually reproducing life cycles. The zygotic in fungi and protists, and the sporic or alternation of generations in plants and algae, and you'll explore that later. Now, let's go back and kind of investigate this idea of genomic structure a little bit more closely. So again, we know that genes are a unit of heredity. We know that they're segments of DNA. That's kind of a molecular understanding. But a lot of this stuff was figured out way before we knew what DNA even looked like. We know that the gametes, the sperm and egg, are what we kind of package that DNA into so that it can fuse to make the next generation. Now, the genes on your chromosomes have very specific locations, right? And they're called loci, plural, or locus, singular. So that's very, very important, because as we mix up the genes, we want to make sure that we don't delete or forego handing off any particular gene. And like I said, as we delve into the mechanisms of meiosis, we'll see how this is important. But we kind of need to get some vocabulary down first. So one set of chromosomes is inherited from each parent. So let's look at this diploid flower, for example. This is one example, just one set of chromosomes it has. And every location on this chromosome has copies of the same gene. Now, what you're going to notice then, though, and this is where variation occurs, is that there's many versions of genes in some cases. So we call these alleles. So the allele for purple flowers is located at the same location on one of these chromosomes as the allele for white flowers. So at that locus is the gene for flower color. And then one could ask, well, which version of the gene for flower color do you have? Well, one chromosome has the purple version, and the other chromosome has the white version. It's the same gene, quote unquote. It's the same protein, right? But there has to be some slight difference. Okay? This is mutation. Okay, mutation allows variability to arise. In this case, purple versus white flowers still serves the same function as giving color to the flower. Okay, so as we think about this gametic life cycle, we have to kind of delve into the types of cells that are going to play a role in the life cycle. So again, somatic cells have 23 pairs of chromosomes. You have 46 chromosomes. You need all 46 for that cell to be functional. I can look at all 46 through a karyotype. We've seen some of these before, where I can literally map out the chromosomes and count them up. 23 pairs, 22 identical pairs, and in this case, this is a picture of a male karyotype because there's an X and Y, and we'll get to that in a minute too. All right. Now, the pairs are called homologous chromosomes or homologs. Right? They're the same length, they carry the same genes, but they may carry different versions of those genes. Right? So this is the idea that you have 46 chromosomes that can be organized into 23 pairs. So we call this ploidy. The number of copies of each gene that you have on those chromosomes, or the number of copies of each chromosome, you can put it that way, is your ploidy. So most animals are diploid, having 2 copies of every chromosome, a pair. There are many plants out there that are tetraploid or octaploid, so you can have different versions. But for most part, we're going to look at examples now that are diploid. Now, if I'm diploid, yet I'm going to hand off only 1 copy of my genes to the next generation, that gamete, that specialized cell for reproduction, is now haploid. It has 1 copy. So 1 copy is called haploid, 2 copies is called diploid. So what this leads us then to is looking at the pairs, how that one set isn't homologous. And so that's going to set us up to distinguish between autosomes and sex chromosomes. So the sex chromosomes are the X and Y chromosomes. Humans have a homologous pair of X chromosomes, yet males do not. Males have an X and a Y. So the 22 pairs of chromosomes that are not the sex chromosomes are called autosomes. And those are always going to be paired up. They're always going to line up and do what we call crossing over later as we learn about the mechanism of meiosis. So each pair of chromosomes includes a chromosome from each parent. So in each pair of your first set of chromosomes, one of them you got from your mom and one of them you got from your dad. So again, this idea of 46 chromosomes is necessary that we can organize in sets of 23. A gamete then would have but 1 chromosome from each of those pairs. So a diploid cell has 2 sets of chromosomes. A gamete is going to have 1 chromosome for each pair, but you need 46 chromosomes to be functional. All right. So again, this is where it gets a little bit tricky when we talked about mitosis. As we get into meiosis, which is going to be the process to make the gametes, we need to keep our numbers straight because we're actually the goal is actually to have those numbers. So I want to emphasize again this idea that when we're going through the cell cycle and we're headed for making gametes, we are going to duplicate our DNA just like we would in mitosis. But that duplicated chromosome is still considered 1 chromosome, right? So let's go back to this slide from our earlier recording. We've got 1 chromosome on the top. We duplicate it. That's called 1 chromosome. It's a duplicated chromosome, but it counts as 1 chromosome made of 2 sister chromatids, which then ultimately will separate, each becoming a single chromosome. All right? So again, the idea that gametes, sperm or egg, are haploid. Right? So now you have 23 chromosomes. All right? And so that is going to be the mechanism by which we avoid continually doubling the amount of DNA in our offspring. We hand it off half, and half makes a whole again. So again, if we look at the karyotypes, here's a female karyotype. You can see all 23 pairs in that last pair, X. You've got a homologous pair there. But in a male, you have that little tiny Y chromosome instead of a pair of X chromosomes. So these are going to line up when they line up on the metaphase plate, but they will not exchange material, as we'll see to that later. Now, determining gender by XY by a sex chromosome isn't the only way to do it. There's an XO system, there's a ZW system, there's a haploid-diploid system. There's all kinds of systems to determine gender. So again, don't get caught up in the idea that because we're using humans and animals as an example, it's the only way that that you can do this. All right, so going back to this life cycle, let's get some final terms in here that we're going to need to kind of bring this all together. So fertilization is the union of the gametes. The fancy word for this is syngamy. Once that sperm and egg are united and now kick the chromosome number back up to 46, it's truly a diploid cell, you call it a zygote. And I've mentioned this before. That is the single cell that most animals and plants, most eukaryotes, start as. A single-celled zygote. And from that, through mitosis, cell division, it grows into whatever multicellular organism it is. Okay? So now the key here is that as that multicellular organism matures, it's going to produce a secondary cell line that we call a germ cell line. And that germ cell line will then go through a process called meiosis, rather than mitosis, that's going to make the gametes. All right? So that meiotic cell division is going to result in cells that have half the chromosome number. Right? So here we can see it again in our gametic life cycle. So you have a mature diploid organism, one male, one female in this case. They have select cells in the testes and the ovaries, the germline cells, that are going to go meiosis to produce sperm and egg. Sperm and egg are haploid. Two haploid cells fuse, fertilization, to make a diploid zygote. That then grows and develops through mitosis to make, again, a mature organism. Right. So again, meiosis produces gametes. Gametes fuse to make a zygote. That zygote then develops by mitosis into a multicellular organism. So there's a lot of vocab there. This is introducing some new topics, some new concepts. Really think about these, and as we go on, you're going to see that these are the foundation to genetics and reproduction. And as we use these terms more, they're going to become more familiar to you.